Vanakkam friends, and this presentation is on seeds of change. We are transforming inducing changes or transforming the seeds in nature genetically, and the mutation breeding in modern agriculture. This is the topic for discussion, and straight away we will come to the main points in which we are going to discuss or mutation definition, types and causes of mutation, mutation breeding in crops, and mutation application and its importance in crop improvement. Examples, that is the list of varieties released by mutation breeding in India, not only in India but also in the world. Important scientists who are involved in mutation research and mutation breeding, as well as the research institutes that are involved in mutation breeding, advantages and limitations of mutation breeding. These are the topics in which we are going to discuss one by one. First one is definition. Definition is nothing but the sudden and heritable change in DNA sequence of a gene or chromosome, resulting in variation that may be passed into the next generation. In plants, mutation can lead to new characters like altered plant height, flower color, seed size, resistant to diseases and pests. These are all the changes that we face in mutation. And now we can see the types of mutations one by. There are three types based on origin. First one is spontaneous mutation. It occurs naturally due to errors in DNA replication or environmental factors. These changes are rare in flower color. And induced mutation, that is artificially created using mutagens like chemicals and radiation. And by this method, the variety, a wheat variety, Sharbadi Sonara, was released by induction of radiation. Based on cell type, there are two types again: somatic mutation and germinal mutation. Somatic mutation occurs in non-productive cells that are not heritable through seeds. And whereas germinal mutation occurs in reproductive cells and that are heritable through sexual reproduction, that is seed. And third is based on molecular nature, the point mutation, change in single nucleotide, that may be in base. And chromosomal mutation, there is a structural changes like deletion, duplication and inversion and translocation of chromosome. And gene mutation, that change occurs in chromosome number, may be example the polyploidy in nature. And the causes and the agents that are involved in mutation breeding or mutation of crops are chemical, physical mutagens and chemical mutation, two types. In physical mutagens, X-ray and gamma rays are used to break DNA strands, whereas UV rays cause thymine dimer formation in the genes. And the chemical mutations are many, and I listed out only few. EMS, ethyl methane sulfonate, that induces point mutation. Sodium azide, the second chemical, and most commonly used are colchicine, which induces polyploidy by preventing spindle formation during the cell division, that may be mitosis or meiosis cell division. And mutation breeding in crops by using some physical and chemical mutagens to develop a new and beneficial plant varieties, and the steps involved are the selection of the best plant material, may be seed or vegetative origin, and those things are exposed to mutagens, maybe the physical mutations like radiation or chemical mutations like EMS, colchicine, etc. And growth of M1 generation, that is the mutation 1 generation, that is the first mutation generation, and selection and screening of M2, M3 generation for desirable characters, and stabilization of uh, the selected varieties through different generations, maybe 7 to 8 generations, and release of variety, finally the mutant varieties. Then application and importance of mutation in crop improved, and it is as told earlier, it creates some kind of traits or the characters, short in stature. Sharbadi sonara in wheat is the example. Disease resistance in sugarcane, red rot resistant variety, Co 8152. Co means Coimbatore 8152. Early maturity, that is noticed in Jagannath of rice variety, and improved quality in terms of protein and other things. Pusa Lal Viruti in cowpea, and seedlessness, that is Goa seedless varieties. The examples of muted varieties that are developed in India in different crops, that wheat, Sharbadi Sonara, by using Kama race developed by MS Swaminathan to improve the grain quality. Then groundnut TG19A having high oil content by EMS, ethane methyl sulfonate, and it is developed in bark, Mumbai by MV Rao. Mung bean, green gram, Pusa 105, bold seeds and early maturing variety by Gamma race method developed at IRI New Delhi. Black gram T9 mutant, short duration, erect plant type by using X-rays, developed by Tamil Nadu Agriculture University. Rice Jagannath, early maturity and semi-dwarf variety to prevent lodging of the rice varieties by X-rays. And the Odisha Agriculture University, that was released by Orissa Agriculture University. Cowpea Pusa Meruti, Pusa Lal Meruti, and improved seed color and yield by Gamma race at IRI. Soybean NRC 37, higher yield and early maturity, Gamma race ICR, and Indian Institute of Soybean Research, Indo, that is a joint venture. Cotton Indo 2, improved fiber quality by X-ray, and associated in association with the bark, the IRI released this variety. And chrysanthemum, various color mutants, flower color variation by Gamma rays by Bark Baba Atomic Research Center, Mumbai. Sugarcane Co 8152, red rot resistant, developed using gamma rays at Tamil Nadu Agriculture Coimbatore. Gingerly sesame Coven mutant for high yield and seed quality using gamma rays at Tamil Nadu Agriculture University. And pearl millet TIFT 23A mutant, male sterline for hybrid use, and by using gamma rays developed at Tamil Nadu Agriculture University. Pigeon P TT5, it's a dwarfness, synchronous maturity, gamma rays using gamma rays at bark, released by ARI. Jute JRO524, early maturing butter fiber by using chemical EMS at the Barakpur, the center. I have forgotten this anyway. I will explain in later. Mutant varieties developed internationally. There are many things in barley, tobacco, Drosophila, and tomato. And important scientists who are involved in mutant research and mutation breeding are internationally. Hugo de Vries is from Netherlands. He coined the word mutation and proposed mutation theory of evolution 1901. And based on his work, Unitera Lamarckiana And Hermann Muller, 19, from USA, discovered X-ray can induce mutation. Fruit flies like Drosophila, awarded Nobel Prize in 1946. Levi Statler from USA, he has pioneered the use of X-ray and gamma rays in plants like maize and barley. And Charlotte Aubuck from UK, he discovered chemical mutagens in mustard gas causing mutation Drosophila. Gustafsson Aki from Sweden developed mutant variety in barley. Dr. M. S. Swaminathan in India, he used mutation techniques in rice improvement. The Indian scientists are many. I can list out I will read the name. Dr. M. S. Swaminathan, Shetty from Bork, M.V. Rao from Bork, P.K. Appaduri from IRI, and Padmanabhan from University of Mysore, and B.M. Chowdhury from Directorate of Oilseed Research, Hyderabad, and Santangi, I'm sorry, Santangi TNA, Coimbatore, it is involved in mutation breeding of sorghum and pearl millet. In Hyderabad, the Directorate of Oilseed Research, they involved in mutation breeding of groundnut and sunflower. And University of Mysore is involved in biochemistry of mutagenesis in plant mutation response. Pika Appaduri, he worked in mutation breeding in pulses and cereals, as well as M.E. Rao. He also developed several pulses and oil mutant varieties. And B.V. Shetty is pioneered in radiation-induced mutation breeding, and Dr. M. S. Swaminathan used techniques in rice mutation breeding, and he is the father of Green Revolution in India. And research institutes that are involved in mutation breeding are Baba Atomic Research Center, Mumbai; IRA, Indian Agriculture Research Institute, New Delhi; Directorate of Oilseed Research, Hyderabad; Tamil Nadu Agriculture University, Coimbatore; Punjab Agriculture University, Ludhiana; Orissa University of Agriculture and Technology; ICR institutes that are leading center for rice and pulses; and Central Research Institute for Jute and Allied Fibers, Barakpur. Regarding advantages, it is a fast method to create variability. It introduces novel traits not found in natural population. It is useful for improving vegetatively propagated crops, that is more important. It is suitable for self-pollinated crops. And limitations are most mutations are harmful and lethal. It needs large population for selection. It is a time consuming to stabilize mutants. Maybe it will take up to 8 or 9 generations. To conclude summary, mutation is a powerful tool in plant breeding to introduce useful variability. By using the induced mutation, and there are about 3,000 mutant varieties are released globally. And regarding India, it has contributed successfully for using gamma rays, EMS, and colchicine for improving the yield and quality resistance as well as adaptability of the crop. Thanks a lot, friends, and kindly subscribe my YouTube channel and give likeness to this presentation. Um, Share this presentation video to your friends as well as the group, WhatsApp group. Kindly comment on my presentation. Thank you very much, friends.